Welcome everyone to today's lecture. Today we have a guest lecturer from Karolinska Institute, Mite Mijalkov. He will talk about brain connectivity and a software they developed for analyzing brain image data. So he works in neuroscience in Karolinska Institute, and he has been also doing his PhD on this topic. So he has wide knowledge of connectivity and brain. So without further ado, I let him start his lecture. Thank you very much for coming, Mita.
Yeah, I could, and also thanks for inviting me and also for giving the chance to give this lecture. It's always nice to be here and to speak about brain connectivity and how these things work. So just give me a couple of seconds in order to share the screen, and then just a second, and then I should be ready to go. Okay, so
great. Okay.
I think it worked, right? Yeah.
Yes,
So
we can see it. So you can start. We hear you perfectly and see it perfectly. Great.
thanks a lot. And as I could mention, I'm going to more than actually showing the software, I'm going to show some results or some things that we found out about brain connectivity using this software, especially in Alzheimer's and Parkinson's patients. And if you are interested more about these topics, you can always email me or visit the software website. Both of them are written on this slide. So the punchline basically is that the brain is a complex network. And this complex network exists at many spatial levels. So when you start from single neurons, then the neurons kind of come together to form networks of neurons that can do some particular function. And then these networks of neurons also come together to form different parts of the brain. In this case, for example, I show you the visual system. That these parts of the brain can actually kind of do their own specialized functions. And finally, these different parts of the brain doing different functions, they can come together and form a whole brain network, which is what we currently have, we currently study, and this is the basis of all of the complex behavior that us as humans can actually do in different type of situations. And historically, this has been studying basically in two ways. And the first way that has been very popular is to study the different parts of the brain and to actually see how different brain areas are specialized for a specific function and what makes them do that specific function. On the other hand, with the current advance of different imaging techniques, which were able to give us much more data about the human brain and how it behaves in different type of conditions, we figured out that studying just specific brain areas is not enough. However, one needs to also study how these specific neuronal populations interact between each other, and this should give a more complete understanding of how the brain works. So the good thing here is that I'm going to speak about the brain quite a lot in this situation. However, the topological organization or the principles that I'm going to speak today are shared among many different networks. Some of them I have shown here. So just by changing a bit of the nomenclature, just changing a bit of the principles, everything that I am going to speak here more or less could be used to study the organization of these different type of networks. So just so that you have a general idea of how this lecture is going to go, I first am going to speak about these imaging methods and how we actually acquire the data, and then explain a bit the process about how we actually use this type of images to basically build a complex network that represents the human brain. And then in the final part, I'm going to show a few works that have been performed that show how the brain networks are organized in healthy individuals, but also how these type of networks are disrupted in different diseases. And I basically concentrate on Alzheimer's and Parkinson's disease as one of the two most common neuronal disorders. So everything starts with the magnetic resonance imaging. And the story of the magnetic resonance imaging is a story of two Nobel Prizes in particular. The first one is the one in physics, which has been awarded in 1944 about the magnetic resonance. And then the second prize was awarded in 2003 in physiology or medicine. And this has been awarded to these two researchers for developing, for let's say, using magnetic resonance in order to perform some imaging. They have done their work independently of each other, and which resulted basically in 1980 to build the first commercial MRI scanner. Of course, we have come a long way since then, and these techniques has been improved in, spatial and also temporal resolution in the case of functional imaging, but they basically amount to one big giant magnet in which you put the person inside this magnet, and then using some different properties, you try to obtain the image. Going into a bit more detail, the human body is built from many elements. However, in MRI, what is most commonly used is the behavior of the hydrogen, because hydrogen is basically a single proton and it has a very distinctive magnetic spin. So in our normal state, obviously we are not magnetized in our normal state. So what happens is that the dipoles of these hydrogens are randomly distributed towards the brain, and therefore we don't get any net magnetization in our normal conditions. However, once a person is put in this giant magnet, what happens is that now some of the protons will try to align with the field. And while most of the protons chose the low energy configuration and they align with this magnetic field, there is some substantial part of these protons that are going to align anti-parallel to the external magnetic field. And one thing that is also good to note at this point is that alignment here doesn't mean that they're static and they just basically follow the field. It means that there is a frequency with which they actually precess, in which they precess across the field, which is called the Larmor frequency. And that frequency is different for different tissues. So just to summarize, is that in outside magnetic field, we have randomly oriented hydrogen atoms, so therefore no image can be taken. But inside the magnetic field, we have a net magnetization due to this alignment of the protons. And the net effect of doing this external field is that there would be a longitudinal magnetization along the field, because an excess number of protons would be aligned with that field. But however, we will have no transverse magnetization or no magnetization in the transversal plane, because all of them precess around the magnetic field with different frequencies. So on average, due to this precession, they would cancel each other. So in order to take the image then, you need to disturb this kind of equilibrium system, and you do this by applying a perpendicular radio frequency field. And this means that if you just send the radio frequency field and this particular frequency with which protons precess around the magnetic field, you could basically excite the protons, and few of them can pass to the higher energy state. So what you're actually doing is that you're kind of increasing the radius of their precession, and therefore you kind of tip or tilt the magnetization into the transverse plane. And the thing here that we use the mostly is that this excitation happens if you send this radio frequency field at a particular resonance frequency, which is actually different for different tissues. So therefore, by sending these external fields at different frequencies, you could basically build the sample of the composition of your brain, and you could create the contrast between the different tissues. And then you basically turn off the field and you try to see how the protons come back to their initial state. And it turns out that they come back into two cases. The first one is what is called T1 relaxation. So this is kind of like when they interact with their local environment, this is how the longitudinal magnetization comes back and how much the longitudinal magnetization is retained after the field is removed. And then you can have like a T2 relaxation constant, which measures basically how much the transverse magnetization loses their intensity, or how much the spin again come to be defaced in that sense. And you can see that if you do this across the brain, you can obtain two different images that can show you the structure of the brain, that I showed images at the below. And therefore, from here, you can kind of assume that doing this will give you the information about the structural composition of this brain. Uh, if you actually wanted to see at the functional composition and how the brain activity changes or brain activation patterns changes during some task that the people would perform, then we use something called functional magnetic resonance imaging, which basically measures metabolic activity. So normally in the brain, you have hemoglobin. And here in this simple sketch, the blue circles represent hemoglobin, which is poor in oxygen, and the red one represents the hemoglobin that is high in rich in oxygen. So if you have a stimulus, or if a person tries to do some task like finger tapping, or he wants to see something, then the theory goes that the part of the brain that is kind of mostly activated or responsible about this task is going to need more energy. There should be more oxygen flowing to that part of the brain. And as it turns out, whether you have oxygen rich or oxygen poor hemoglobin, both of them have different properties when put into magnetic field. So therefore, by using this type of property, one could actually measure the difference between the oxygen-rich and oxygen-poor hemoglobin, which would give you an indication of which part of the brain has been activated under this stimulus. And therefore, the fMRI here would give the information about how the different brain regions co-activate together by using this type of analysis. So the way that you basically do is that you divide the brain into many voxels, and voxel is basically a three-dimensional analogy of a pixel. And then what you do is that you perform multiple measurements over time. And you basically scan the whole brain over time, multiple times, and what you obtain is a one-time series per a voxel, which basically tells you how this voxel activated over time as a response to some task. However, not only task, but fMRI is basically taken in the resting state, where patients are in general instructed to lie down, and people just take the spontaneous activity of their activation in the brain. And on the other hand, people sometimes are put into task-based paradigm, where you have blocks of rest with and then you have blocks of some activity, which is, for in this case, finger tapping, and therefore you are going to see which voxel are responsible for that activity, or in this case, like which voxel are activated while doing that particular activity. So this was just a very brief summary about how to take the structural and the functional activity of the brain. And now I'm going to speak about how actually we take these images, which don't look at all like complex networks, and how we basically extract this information that we can use then to analyze how the brain behaves. So from the previous courses that you have had, it's basically have been shown to you that this is a complex network. And this is a very simple visualization in which you have a network which consists of nodes and consists of edges. And then you have graph theory, which is a powerful mathematical framework which can analyze this type of complex networks. So in order to go from this image on the left to this network on the right, there are few things that needs to be undertaken so that everything is done properly and different patients could be actually compared to one another. And the first one is basically we need to define what the nodes of this network would be. So we need to say what the nodes represent. And in this case, we will take the nodes to represent the brain regions. So we end up here with networks which are 200 or 800 nodes. So therefore, it would be computationally infeasible to basically have a node represent a neuron that would just not be calculated, or nothing could be calculated on that network. And also we don't have the available data in order to do that thing. So we have to kind of group the different voxels together and have an extended region represent a single node. So simple considerations here are that the nodes should cover the whole brain, and you need that the nodes should not overlap with each other. So you need different brain areas to be different part of the graph. And luckily for us, a lot of research has actually been done on this. And now there are very good ways to define the nodes of this network. And it could be data driven, where you just have a few scans and you see how your data separates and which nodes kind of are function together. So then you take the regions that function together to be your node. Or you can use some anatomy of the brain, anatomical knowledge, and you say that brain node, like brain regions at different parts, will constitute a different node. So after we have defined the nodes of this brain, the next step is to basically define the edges. And here it's where it gets a bit more tricky, because depending on the type of measurement, you actually have different ways to build these edges, and you have different ways to actually also interpret the network and what does it mean. So the very simple thing that we are trying to ask is that what is, like, how strong is this connection shown in the bool? And one way to do it is to look for structural connectivity, to see the physical or structural connections between the brain regions and see how strong two brain regions are connected structurally. And the way that you do it, it's basically you, after you have taken your scan, you measure the cortical thickness around each region. The resolution here is not good, but you see here that after the cortical thickness is the thickness of the gray matter that kind of goes to that region. And this basically assumes that if you have couple of regions that are performing the similar function, then these two regions are expected to have a similar structure around them. So that's the core of this structural MRI network building. And then basically you obtain one value per region, per subject. And then if you have your subject in a given group, you can correlate the values of the regions across that group. And therefore you obtain a certain Pearson correlation coefficient in most cases. And this Pearson correlation coefficient is an indication about the strongness of the structural connection between those particular two regions. The second way to define the edges is if you have functional MRI. And this is a bit tricky because this is not a physical connection. Instead, the functional connectivity measures a statistical dependence between the signals that are measured in the functional MRI between two brain regions that are far away from each other. So as you can see here on this small sketch, you have two regions. For each of the two regions, you're going to have an associated time course of their activation pattern. And then once you calculate the correlation coefficient between these two time series, you can get an estimation of, in this case, functional connectivity between these two regions. And then finally, there is this type of connectivity, which is called effective connectivity. And this basically measures the influence that one neuronal system has over another. So this is a type of directed connectivity where you are looking for causal relation, so that if one system activates first, what effect does this have to the rest of the brain in their activation? And here I should also mention that the structural connectivity and functional connectivity generally don't give any indication about the direction or the influence or the cause of that interaction. They just tell you whether that interaction is there or not. So after we have done this, either we have done the nodes, we have defined the edges, we have performed these pairwise correlations for all pairs of regions, then we can collect everything together, all pairs and correlations, we can collect them into the adjacency matrix, which is now the network that is representative of our brain. And one thing that I would like to mention here is that the adjacency matrix by definition is weighted. Because person correlation is a continuous number between -1 and 1. But two things are very common to do here. One thing is to set all of the negative connections to 0. And this is just because of some interpretational problem with the negative correlations. It's not very clear how to interpret them at this time. So what most of these analyses do is that they just neglect the negative correlations. And also, instead of analyzing the whole weighted networks, what one could do is that you could threshold this type of network, and therefore you can say that your edges will be 1 above a given threshold and 0 below, and then you can analyze this binary network of 1s and 0s. You can analyze its topology. So then once you have had your networks, you put your subjects into groups, you have your healthy controls, you have your patients. And then one thing that you would like to see at this moment is whether there are differences in certain topological measures between these two groups, and whether we can actually find something out from these networks. So one good way to do this is to do permutation testing. And this is a statistical test where you first calculate a given measure for each group. And the measure here could be global efficiency, could be a path length, could be clustering in that network. And then you calculate the difference of the average of this measure between the two groups. And then this permutation test would essentially test whether this difference is different of 0. So whether you have non-zero significant effect. So once you have this difference, you need to ask yourself whether this is a significant difference. And what you do is that you can basically mix the group. So you randomly assign some healthy controls into the patient group and vice versa. You recalculate the measure, you recalculate the difference. You repeat this many times and you get a given distribution of these differences, and each difference, each point in the distribution corresponds to a single instance of a permutation. So then if you have a situation like this, this would imply that the p-value is very high due to this black region here having substantial number of measurements. You would conclude that whatever you observed, it's not significant. But for example, once you have repeated this many times and you get this type of distribution, this would tell you that you have a very high p-value, which would give you a significant result. So this is one thing that needs to be done generally at the end of the analysis. But another thing is we need to account for things that we have in our experiments, but that are not important in the experiment. So these are not the quantities that we look for, but these are quantities that can affect our results. And for example, Parkinson's disease is a movement disorder. So then Parkinson's patients can actually move quite a lot while the MRI is performed. So therefore you can have misaligned images as you take these images across time. Or for example, it has been also shown that in Alzheimer's patient, the number of years that this patient spent in education and in studying could actually have influence to the deterioration of their networks. So we have to account for this motion and for this education, but we are not really interested in that. We're just interested whether the topological structure of the groups of the networks is different between the two groups. So what happens is that we define this motion and education, and also here we could mention that gender and different like right-handedness, left-handedness, and these kind of things, they're all covariates in our statistical analysis. So what we do is that this is like just a sketch to show you. So if you plot your value of interest on the y-axis and you plot the covariate on the x-axis, you might get something like this, which would show that there is a kind of correlation between what you want to observe and the education. So what we generally do is that we fit a baseline to this type of data, and then we find the residuals or the deviations of our actual data points from this baseline behavior. And therefore, what you do is that instead of basically doing the permutation test on the original value, you actually do your permutation testing on these residuals, which are then the values when the effect of any covariates is kind of regressed out. And this type of analysis should give you the statistical effect when every nuisance or covariates are accounted for. So this was also the second part, where I also summarized a bit how we go from an image to a complex network. And then now I have come to this final part, which consists of few studies that I'm going to speak about, which are going to give some indication about how the brain and networks look in health and disease, and what we can actually learn about this type of behaviors. So one thing that I need to basically say at this moment is that all of these things that we have discussed and all of this graph theory that deals with the networks assumes that the networks are in topological space. So basically it assumes that the actual physical position of the nodes is not really important. Only the interactions between the nodes are the ones that kind of designs how the network is going to behave. On the other hand, we have the brain, which is obviously a structure in a physical space. It's every single node is placed at a different part of the brain. So therefore you have, like, in addition to actually the topological information, which can be kind of accounted by all of these neuroimaging techniques that I was speaking about, you also have the physical location, the physical distance between the nodes that can also affect your results. Now, one thing also to say is that while it seems that the physical space is important, there is some interesting caveat here, is that most of the networks who are actually, again, can be taken to be like in a physical space, it has been shown that actually it's their topological performance that is kind of more important when you are trying to kind of infer how the network is going to behave. Like, for example, in protein networks, you have this topology is kind of determining whether some functions of this metabolic and gene regulations. Then you have the social networks. And while the social networks, for example, if you look at the social media, they are in topological space. But if you look at the social network from a friendship point of view, you have a lower probability to be friends with someone that lives far away from you. However, it has been shown that the topological interactions are kind of more important in this case. And also you have the power networks, where like, while the power is maintained by different structures that are placed in a physical space. For example, there has been few studies on the blackouts in these power networks, and it has been shown that not the physical space, like the physical distances between the networks, but however the connections in the electrical grid are the ones that are more important. So although the brain is in physical space, topological properties of the brain networks are going to become very important for these things that I'm going to speak about. And they are, I'm going to basically discuss now that this topological organization is going to be related to the robustness of the brain network to perform different functional tasks and to actually adapt in different cases. So I didn't speak about graph theory just because Giovanni and I could have covered this in the previous lectures. But here in these slides, I'm just going to pop up few things just to as a reminder so that you could have a better idea of why the things that I am saying are in that way. So if you remember, the clustering coefficient measures the number of triangles or the fraction of closed triangles that exist around the node. And in the sketch on the left, you can see, for example, that the red node has a higher clustering coefficient, while the blue node has a low clustering coefficient when compared to the red one. And also the modularity or the community structure of a given network is that basically you look for a set of nodes that are very densely connected with each other. However, they have very less connections between different communities. And now, in order to go this and to apply this to the brain networks, it has been shown here that local connections, having a dense amount, a high amount of local connections, is very important for specialized tasks. And this is because anatomically, brain regions are assumed to be very near to regions that are going to perform this specialized task together, and therefore they would have a lot of connections between each other. So therefore, a specialized brain regions has been associated with a higher clustering coefficient. This again, as an example, we could give the visual processing, and we could say that the regions in the occipital cortex, these few regions that are present there, will have a lot of dense connections between each other, which are going to actually be involved in the visual processing inside the brain. Now, another let's say structure which is very important for the brain for the localized task is a well-defined community structure. And then you could imagine here that these densely interconnected nodes that are all in the same module would actually be connected together in order to perform better that specialized tasks. And therefore, we come to the conclusion that nodes in the same community would perform similar functions. And actually here on the bottom right, you have a sketch of a study that I took. It's a kind of mapping of the community structure in the human brain. And you can see that there are distinctively 3-4 communities in the brain that are going to be associated with different cognitive functions in that case. Now, while local connections were important for these specialized tasks, we already know that the brain needs a way, like that the different regions in the brain communicate with each other. So therefore, we need to ask ourselves then what is important for the global interaction between these type of regions. So here, I would like to remind you about the shortest path length, which is basically the shortest distance between two nodes. So even though there are many different paths that an information could take in order to go from one node to another, in this case, a blue path and a red path, the red path is 3 and the shortest path length of this communication is 3, just because we assume that the information will always take the shortest path available to it. And in this case, shortest path here means the number of edges in topological sense that needs to be passed in order to these two nodes to be connected. And then if you just inverse the shortest path length, you obtain the global efficiency. And these two, obviously these two measures are very closely connected with each other. And they kind of pertain to the same information, which is important for the global integration. And it has been shown in actually many studies that a better integration between brain regions is facilitated if that network is filled with paths that are of low lengths or high efficiency. And one study actually measured the IQ scores of the healthy volunteers, and then they tried to see how they are connected with the topology of the network, and they figured out that these high IQ scores had a negative correlation with the path lengths, both in structural and functional networks, which basically tells us that topologically, if you have shorter path lengths in the network, the information will actually be transferred much better during your brain network, and which is associated with having a higher IQ in that case. So all of these things put together led to an interesting theory about how the human brain connect on networks are built and why they are built in this way. And this can go under the name of the economic theory of economics of the human brain. And basically this means that the brain basically operates at the same principles as a normal market would operate, where we would like to basically maximize the profit while controlling for the cost of our products, whatever we do. So basically, this type of networks would work as a trade-off between the costs that you have in order to build this network and actually the market value or the processing, the efficiency of processing of an information in this network. And everything comes back to Cahal, who has actually done a considerable work in this type of brain organization. And this quote in red is quote of Cahal, where he basically says that all of these principles of operation of the human brain can be considered to be governed by the laws of the conservation for the time, space, and material. And then we need to kind of conserve all of these three more or less simultaneously in order to kind of get this type of brain organization. So if you think about time, conservation of time basically means that you try to conserve the information transfer, or you actually want your brain to be built in such a way that the information transfer between two spatially different brain regions are going to occur in the shortest amount of time. When we talk about the space, space is basically connected also with the material here. And these two, space and material, basically refer to the wiring costs or the metabolic activity, the energy required and the material required to actually create a short or long distance connections. So here we are trying to figure out whether our brain would be better served if he has a lot of short connections, or whether the long connections are better to be done in general. And also, since our brains they fit within the skull, so we have like a limited volume where the brains needs to be actually fitted. Here, the considerations of the space and material also account for how many connections physically you can actually have into the limited volume that is occupied by the brain. So it has been this brain organization has been postulated that is kind of like an optimal trade-off or a balance that kind of tries to control for these three different types of considerations that we have. And I see that the time is 10:40 now. I think that this is a good time to actually take the break, and we could meet here. I will continue the lecture at 11, so that I will continue and to show you how these conservation principles actually were able to, by considering these conservation principles, how we are able to observe and learn about the organization of the brain networks that we know at today. So see you in about 20 minutes in that case, and have a nice break. Okay, so let's continue with the second part of today's course. And at the end of the first part, we were discussing about Cahal and his conversation principles of brain organizations and how the conservation of time, space, and material actually drives how the human brains are formed. And we know about it today. So first, let's look at the conservation of material in a bit more detail. So many studies have basically shown in the concept, in the framework of structural connectivity, that cortical neurons that are actually close to each other, they have higher probability to form synaptic connections between each other. And also, if you look in the whole brain, and if you look at the connection distances between the brains, you would see that this distribution of connection distances is skewed towards short distances. So short distances have a greater chance of occurring in the human brain. On the other hand, if you look at the functional connectivity, you can see that functional connectivity, the strength of the functional connectivity between two brain regions decays as an inverse square of the physical distance, which basically means that the further you are, the more chances are that you have less structural, less functional connectivity with each other. And finally, if you remember that you could threshold the connectivity network, so once this connectivity network has been thresholded at sufficiently higher thresholds, it has been noticed that the most surviving edges, they actually have a relatively short physical distances, correspond to these short distances. So therefore, we see here that the basic principle of this conservation of material is shown in such a way that short distances have very minimum varying cost, and therefore they tend to occur more into the brain. However, if this was the only principle that our brain was built after, we would actually end up with a lattice network, with a regular network, because as you can see here, every region is connected with the neighboring regions that are very close to each other. So therefore, you would have a lot of connections made up only from neighboring regions. Now, if you go and discuss the conversation of time, you would see that the main aim of the brain is to basically facilitate quick integration between any two brain regions, and this is facilitated through smaller topological paths. And therefore, here we see that short paths and actually directed connections. So instead of taking like 5 or 10 edges in order to reach two regions, if you take 1 or 2 edge, is preferable if you want the time to be conserved. And we see that if our brains were actually taking only this road to conservation, we would end up with a random brain network. However, the problem is that none of them can be the only way that our brain is built. So not only one of them can be actually conserved. And one of the reasons is that, for example, since we have to pack a lot of neuronal cells into this finite volume, all of these projections, all of these neuronal cells, they cannot be straight line. At some point, they must be perturbed from their minimal cost, and they have to go to curvy lines just so that they can fit in the finite volume, which already tells you that you can not only minimize the varying cost, but also if you go to the functional properties of network, sometimes if you have two regions that are very physically very far away from each other, but they make a single edge between each other, they actually have a better way of communication. So therefore, at some point, the topological distance is actually more important than the physical distance. So we cannot just go ahead and conserve the physical distance, because that would kind of mess up with the functional properties of the network. This is not for humans, but this is basically for macaque and C. elegans, the worm. And the worm is also one of the few species for which all of the connectome has been very well defined and very well built. So you can see here that in blue, the study plots the total wiring length, so the total length of connections in the structural connectome in the macaque. And you can see that it goes around 50 millimeter in total. However, what they did is that by using some statistical methods that I will not go into, they figured out that the same connectome can be rewired, and therefore you could get some similar connectome with much small total wiring length. So if we were, and also that goes for both species. So if we're just going to conserve the wiring connectome, it means that the one in red would have been preferable for this type of species. However, we see that that's actually not the case, because if you try to optimize the total wiring length, you actually end up with larger average path length. This basically means that you end up with less efficient connectome, which means that the connectome in red is not preferable in this case, although it does have the minimum wiring length. Similarly, for humans, they plot the global efficiency of the connectome as a function of the percentage of the connections that are present in the connectome. And they obviously saw that the more dense the connectome, the better the efficiency is, because the more connections you add to the brain network, you get the probability of basically having a single topological edge connecting two regions. So you are actually kind of increasing the efficiency of that connectome. But when they plot the efficiency minus the cost, which they call the cost efficiency, and then normalized it so that it's between 0 and 1, they saw that the connection density increases up to some point, and in this case it's 15%, and also the efficiency and the wiring length can be both optimized, but then the efficiency kind of decrease with the connection density. So even though more connections are preferable to basically optimize time, they're not preferable to basically optimize both time and wiring cost. So at the end, evolution had it that our brain end up with an optimal balance, which is called the small world network. And this is basically, it takes the whole properties of the random network, where you get the short path lengths, so the high efficiency of that random network, but then it also includes some, so it took this random network, it took also the regular network, the short path lengths or the short distances, if you wish, and then it created the small world network where most of the nodes have similar degrees. However, you have some nodes that have very high degrees in order to facilitate better information. So therefore, the cost versus the time efficiency optimization is kind of in a very well-guided balance. And now we actually know that the small world network actually has very high clustering. So this means it's a very good optimized for local functions, for localized functions. It also has a high global efficiency, so it's very well optimized for the integration or let's say information passing. Then it also has a modular community structure, which again, it's kind of like optimization, because the different communities would be performing a specialized functional task, and then they would be connected with few regions with long-range connections, which are actually the hubs of the network, and these hubs would actually facilitate quick and efficient transformation between the different communities. And then we again see that this hub pop up in the heavy-tailed degree distribution, where you have most of the regions that have similar degrees, similar to the random network. Most of the regions have similar number of connections. However, you have these specific regions which make very large number of connections, which are considered the hubs in the network, and they are the crucial ones that are making our brain healthy and functioning with a good high efficiency. And one interesting thing is that depending on the various cognitive demands, the brain can reconfigure itself in around 10 milliseconds or so. So basically, if you have less demand, the brain can go to a different state. But if you have more cognitive demand, the brain can kind of morph between regular and random network depending on the strength of the task that one needs to do. And this degree distribution is again shown in this graph, where on one side you have the random network, on the other side you have the regular lattice, and then the human brain is somewhere in between, where it takes the degree homogeneity to 1 degree, but then it also has some nodes that add heterogeneity in this degree, and you have some hubs with very high degrees. And you can see here also in the sketch in the middle of the human brain that there are a few nodes that really make a lot of connections when compared to the other ones. So this was a short summary of how the healthy brain works. And if you remember the discussion of the first class, this healthy brain can be measured, can be imaged. And the two, basically the two frameworks in imaging are the structural analysis that you see on the left on the screen, where you get your anatomical parcellation, you take an MRI, you build a structural brain network per group, and then you analyze this brain network using some graph theoretical measures. And also on the other hand, on the right, you have the functional framework, where you actually take functional time series data for a region, you build functional networks, and then you again use graph theoretical analysis to analyze these functional networks. So now I'm going to show you two studies, one for Alzheimer's patients, which is about the structural connectomes, and one of Parkinson's patients, which is considering the functional connectomes and how these changes occur in this particular disease. So in this Alzheimer patient study, we basically compared healthy patients, the connectomes of healthy patients, to patients that have mild cognitive impairments and patients with Alzheimer disease. So mild cognitive impairments is a stage between healthy and AD patients where the AD is not yet diagnosed, but some cognitive impairments are noticeable in these patients. And this is a very shortly to remind you that, the shortest path length is the steps that you need to reach two nodes, that is averaged over all nodes. The clustering coefficient is basically the nodal clustering coefficient that I was explaining earlier, averaged over the whole nodes. Transitivity is something similar to the clustering coefficient. It measures the same thing. And modularity is how good a given network can be segregated into communities, just as a reminder, because this is what I'm going to show you now. So we have basically seen here, what you're seeing basically is that the ones in the gray are the 95% confidence intervals that are calculated by permutation test. And the orange line basically gives us the difference of the given measure between the controls, between the sick group and the healthy group. So in the shortest path length, you see that the patients with Alzheimer's disease and mild cognitive impairment tend to have higher shortest path lengths. They also tend to have a lower clustering coefficient. If you look at the bottom right on your screen, you will see that the Alzheimer patients tend to have a lower clustering coefficient. Over all densities, and this is a binary analysis, so that's why we present our results as a function of density. So overall densities, you see that the sick groups also have, the patient groups also have lower transitivity, and they have higher modularity. So finally, there is the small worldness, which is not very different between the two groups. So this means that these kind of changes are not very well defined and cannot be very well detected. So now let me go back and just very quickly interpret these results and tell you what does this tells us about Alzheimer patients. So if you go with the shortest path length, you see that it increases, which basically means that the information transfer within the network of these Alzheimer patients is disrupted. So you have less efficient connectomes. If you go to the clustering coefficient, you see that it decreases in the patient groups, which basically means that the connectomes of the patient's groups are less specialized or less efficient into do specialized tasks. The same conclusion can be drawn by the transitivity. And here we see that the increase of the modularity basically means that there is some kind of disturbance, like the connectome, the structural networks of these patients are much more fractured. They are more divided into communities more than it's actually normal for the balance of the small world networks. So therefore, you see you have a fractured network, which is less efficient to do specialized tasks, but is also less efficient into integrating information between the different parts of the brain. And this is one hallmark of the Alzheimer disease, which is kind of thought of as a disconnection syndrome, where you have a complete disruption of the structural connectome, and the connectome doesn't work as efficiently as the one of the healthy patients. Now, in the second study, I'm going to show you some functional imbalances in Parkinson's disease patients. And for this, we have been used a cohort which is freely available from the Parkinson's Progression Markers Initiative, which consists of total of 95 Parkinson's disease patients and 15 controls. So first of all, let me tell you a few steps about how we built this connectome, because it's a bit different than the standard way of building that I have been speaking about before. So basically, everything that I have spoken about in terms of functional connectivity assumes that the brain areas co-activate together. So there is no delay at all. Everything activates at the same time. However, that cannot be true. There are different physical distances. The areas are distributed across the brain. They have different topological differences between each other, different ways of communicating. So it's very logical to assume that, or to know that different regions will actually activate with a different temporal delay. So if you have, for schematics, I'm showing you this only on 5 regions, but then our connectomes are built from 200 regions in total. So if you have your fMRI and you have measured this activation time series, then since we already know that there is different temporal delays between the co-activation of different regions, that what happens is that we say that instead of doing this calculation as delay of 0, as it's normally done, let's do it as a delay of 1, which basically means that we calculate pairwise the Pearson correlation coefficient with a temporal delay of 1. And here 1 refers not to 1 second, but refers, because fMRI is taken in a discrete time intervals, let's say every 2 seconds, 1 refers to the measurement taken at 1 point after. So these discrete time intervals is the one that we measured. And here, for example, you can see that how the network of these 5 time series looks if you calculate it at a temporal delay of 1. And here you see the corresponding network. So in this case, we are now not building undirected networks anymore. We are actually building a directed networks because now we have a temporal precedence. So we see that the region that activates first is going to be the origin of that connection, and the region that activates later is going to be the end of that connection. So that if a region 1 activates earlier than a region 2, you basically have a directed connection from region 1 to region 2. This gives us the flexibility. I have shown you here on the left the network of delay 1, but you can imagine that you can do this at many different delays, and you can actually have a kind of dynamic functional connectivity where you can see how the connections evolve in time. And this could again be good because you would actually see which times are important for the Parkinson's patients, especially if you have task-based fMRI. You could basically have this nice evolution and see how that works. But now for our discussion, I will just keep on delay of 1, just to keep it simple and to show you just some intuition about this type of result. One interesting thing about this network is that a directed interaction is evaluated in both directions. So you could calculate the directed interaction that when 2 is shifted with respect to region 1, but also you can calculate the direction vice versa when you assume that 1 is shifted with respect to 2, And then if you have a real directed interaction, one of them will be high and one connection will be low. However, this doesn't change the fact that in this matrix, you basically have both of the connection available, and you can see how it works. For example, if you see the interaction between 4 and 5, you see that 4 to 5 has a high connection, but 5 to 4 has lower connection, which means that in reality, the directed interaction should be between, like, originating from 4 and going to 5, to region 4 to 5. But the problem in this is that when you come to analyze this, you have just too many edges. You don't know which are correct, you don't know which are not correct, you don't know which are spurious, so you need to correct and you need to do some auto statistical analysis for that. Instead, what we decided to do in this study is that every square matrix can be written as a part of an asymmetric and symmetric matrix. So if in the first part we had lagged correlation matrix, we splitted it between antisymmetric correlation and symmetric correlation. And here you can see that the antisymmetric correlation is basically a single matrix that gives you the effective interaction. So now you see that 4 to 5 indeed has a edge in that direction, but however 5 to 4 was probably a spurious edge. So it was not included in this analysis. So in this case, you have highly directional network. In symmetric correlation, you basically have a symmetric network, and you just neglect the direction and you just calculate different connections at different temporal delays. And this really is a very effective method to represent the directional flow because you have a very good directionality character of that. And again, as I was saying, is that here we have a single connection kind of summarizing the whole interaction between the two regions, and therefore this should be better to analyze and it should give us some more sensitivity in the analysis. And then this is the summary. We have 4 methods. The bottom you see the conventional method of zero lag correlation between all temporal regions. And here we have the 3 methods that I have been speaking about in the past 5 minutes or 10. And then this is how we are comparing. And we want to highlight the effectiveness of our method, which is the antisymmetric matrix. Before we do that, one thing is that we could calculate this matrices, I told you, at many different delays, and you could go up to the delay of, the number of time points that you have divided by 2. But then the point is that not all of these delays will be physiologically meaningful. And what we have figured out here is that once you plot the distribution of connectivity as a function of delay for different delays, you're basically seeing that for large delays, you're getting very narrow distribution, which means that large delays, basically, most of the nodes make similar functional connections, and therefore large delays doesn't really tell you anything about the variability in the directional character of these networks. So therefore, our study was restricted between delays 1 and 7, which were the ones that were giving enough spread in the correlation weights so that we were able to investigate it. However, in the rest of this part, I'm just going to show you the results for the delay 1. So we have evaluated the standard 5 global measures, and we have evaluated the directed equivalence of these measures. So now we are not speaking about undirected networks as it was for Alzheimer patients, but we are speaking about directed networks. And you see here that, again, it's the same. The blue lines gives you the confidence intervals, and the orange dots gives you the differences. And you see similar results in a sense that the functional connectome of the PD patients tend to be disrupted. So therefore, the global efficiency and the global specialization, the need for specialization in PD patients is actually diminished in that case. And similar results were obtained at all delays that we considered, although I show here only the delay for 1. What is interesting is that here we were able to check this type of correlations between the measures and the results that these patients obtained at different clinical test scores. For example, what I'm showing you here is the correlation between different measures with the UPDRS test scores, which is a standard test which is administered to patients in clinical setting. And generally, the higher score means that the Parkinson's disease has more progressed. So here you see that there is a high correlation between the measures, the topology that we obtain, and the test scores that these patients have on this UPDRS. So here we could make the connection that the topology of the connectome is kind of like an indicator of the progress that the disease has in these Parkinson's patients, in a sense that if the connectome is more disrupted, if they score more on the high test, so the disease is more progressed, then the connectome is more disrupted, and that can be measured by these 5 measures that I have been discussing until now. And again, not only with this test, but there are different tests that measure executive and memory deficits in this type of patients. And we see again that all of these global measures showed this high correlation also with this type of deficits. And another interesting thing is that all of these changes were observed only by the antisymmetric method, which basically gives us the indication that now looking at the directed connectomes in comparison to the undirected one could actually be beneficial in the clinics, because you would have a more sensitive method that could actually capture early Parkinson's disease that could maybe then potentially you could be able to basically treat it better if you catch it earlier. And this is one of the ways that you might be able to do it. And now for my final study that I'm going to explain in the next 10 minutes, 10 to 15 minutes, is a different type of study where instead of looking at the brain at the whole, so instead of looking at the whole brain networks, we're actually going to look at dendrites and simple neurons, dendrites, and the spine distribution on the dendrites. So you see here on this picture, it's a confocal image of pyramidal neurons with and without tau pathology. So the ones that don't have tau pathology accumulated in the top are in the top, and you see here the red ones inside these neurons is the tau pathology that is accumulated there. So just so that we're on the same page, tau pathology here means the accumulation of a tau protein. So this protein can accumulate on the dendrites, can lead to the destruction of spines, and this leading to the destruction of spines, this has been considered as one of the hallmarks of the Alzheimer disease. That if you have the Alzheimer disease, these tau proteins disrupt the normal functioning, disrupt the spines, who are not now able to make very good connections between each other. So therefore, you disrupt the normal functioning. So now we go to the very tiny small spatial scale of single neuron level, if you wish. So in this case, we again are going to use graph theory in order to see how these tau proteins are actually affecting the dendrites and affecting the formation, the presence of spine. And in this case, this is a simple dendrite. You can model it as a cylinder, and then you have certain spines coming out of this cylinder. However, in order to be able to model this as a graph, we make a projection of it and we get on the 2-dimensional plane. So you see these 10 spines on the top, how they're projected on this plane below. And here it's going to be a bit different than my previous studies, because here we're actually, as a measure of the strength of interaction, we're actually going to use a physical distance. So we are going to assume here that if 2 spines are near to each other, they can communicate with each other better. And if 2 spines are far away from each other, then the interaction between the 2 is not as very efficient. So in that case, we are going to develop a strength of interaction, which will be basically the inverse of the distance between the 2 spines. So basically the strength would be 1/d, shown in this figure. And you can see here a nice schematics that kind of summarizes the graph that was built from these spines. And this graph, basically, you can see that it goes from red to yellow for small and large distances. And you can see here, because the spines are ordered from 1 to 10, you see here that you have this transition from red to yellow. So you have spines that are short, near to each other, and you have spines that are larger, like on a larger distance between each other. And you can see here that the strip here around the diagonal is basically telling you that any neighboring spines are generally tend to be shorter, nearer to each other with respect to the rest of the network. So what can we look in this type of situation? Well, we can first look at the community structure. And here it's a very simple explanation of the community structure, where you have these spines that are kind of clustered together. They form a certain community, and then you have different spines form a community, and then you have the third community here formed. And this is an actual plot of an actual dendrite in 2D. So you can see here how the communities are along the dendrite. So what we calculated in these communities is basically the community size, which basically means like how many spines are actually clustered together. And we also calculated something which is characteristic length of this community, which basically tells you not how many spines, but it tells you like the extent of the community or how big the community is within that dendrite, because as you can see here, not all of the communities are the same. And this characteristic distance gives you the, in general, an indication of the extent of this community. The smaller characteristic distance, the smaller the community is. And then again, the number of spines that are in the community is given by the community size. And here we see basically that the tau positive group, they have smaller communities which are shorter. So therefore, it means that the tau positive groups, once the tau attacks the dendrite, it tends to remove spines in such a way that the resulting spines are basically disconnected clusters of spines scattered along the dendrite. So this is a very bad thing, because then you have disruption in the functional capabilities in the neuronal level. Another thing that we can check is the number of spines that are present in these dendrites. And here we again see the tau positive group has smaller number of spines, which means that when the tau protein in Alzheimer's disease attacked these dendrites, it basically was able to destroy some of the spines that are there. And then we have the grouping coefficient. And this is very similar to the clustering coefficient in a sense that this grouping coefficient will give you the probability that a spine would be near to two other spines, one on the left, one on the right. And it just gives you the probability. So the higher the grouping coefficient, the higher the probability that the spines will be tightly squeezed. And again, here we see that the tau positive dendrites, they have higher grouping coefficient, which fits very well with the previous results, in which we say that once tau protein attacks these dendrites, it results in a very isolated, very tightly packed kind of bunch of kind of groups of spines. And then in the final case, we wanted to see a physiological mechanism. So how does the tau needs to attack these spines in order to produce this type of topology? So first, we thought that let's try to see whether tau comes and attacks one spine, destroys it, and goes away. Whether it comes, attacks another spine, destroys it, and goes away. We see here that if you calculate the change between the healthy dendrite and the attacked dendrite after the tau has attacked, you see that the change is fluctuating around 0. And if you plot here for each of these 11 dendrites that we compared, if you plot, you see that this line, the fitted line, is very near to the theoretical line of the healthy dendrites. This tells us that basically tau protein doesn't attack single spines. Then we said, what happens if it attacks blocks of 3 spines? So tau protein comes, attacks one spine and the one on the left and the one on the right, removes all 3 of them, then attacks another, then attacks another. And then we see that if you attack a lot of spines, you are starting to get, so we started from healthy spines, we attacked them, and we ended up with a smaller clustering, with a smaller grouping coefficient, which is what we already observed in the diseased spines. And also you can see here that the grouping coefficient is lower for all dendrites on average. So this tells us that this attack is plausible, that this is a plausible mechanism of how tau dendrites in Alzheimer's disease actually attack the spines. And then in order to double check it, we went there and we continued this thing when we attack blocks of five spines. And we saw even striking difference where once you attack in here 30 groups, so 30 times you attack 5 spines, you see here that the clustering change is really very noticeable, and that the topology of the healthy network has been destroyed in order to resemble the topology of diseased network, of diseased dendrite. And this can happen if you attack blocks of 5 or 3 spines. So the take-home message here, and this is the same for the communities, where again, by doing these attacks, we were able to verify that if the tau protein in the Alzheimer's disease attacks single spines, then it doesn't create any change in the topology of the network, and therefore attack single spines is not a plausible mechanism. However, if the tau protein, if we consider that it attacks 3 or 5 spines, and then we have done this for different blocks of spines, and we figured out that 5 and 7 spines are kind of the optimal. But then the take-home message is that the tau protein attacks these spines in blocks, and by doing that, it can turn, the simulations show that, it can turn the healthy dendrite network into a diseased dendrite network, which can be a plausible mechanism of how Alzheimer's disease occurs in patients on a neuronal level. So these were all the studies that I wanted to show you for today. And just here at the end, I would like to mention that all of these studies that I have been speaking in the last part have been produced. All of these results have been derived by using BRAF. This is a software that we've wrote in order to do this type of network analysis. And it's a combined effort. It's a collaboration between my group. So I work in the group of Joanna Pereira at Karolinska Institute. So it's a combined effort with our group and Giovanni's group at the University of Gothenburg. And we are now working, and very soon, in a couple of months, we are going to release the second version of this software, which is going to do, it's going to have a lot of improvements over the first one, which was published around 3 years ago. So with this, I would like to conclude this lecture. I would like to thank you for being here and for listening to this talk. And if you would like to have any questions, you can either unmute yourself now and just ask me directly, or at the beginning of these presentations, you have my email, so you can always just email me in this type of presentation.
Thank you very much, Mite, for the nice talk. If there are any questions now, you can simply unmute yourself and ask, or write it in the chat. Yeah, there were a few people commenting that they enjoyed the lecture. Thank you very much for coming again.
Yeah, thanks a lot.
And
thanks a lot for the comments.
I let everyone know that we will post these slides and talk on YouTube, slides on the canvas. So you can go and have a look at the contact later if you have any questions. So you can ask Mite later as well.
Definitely. Okay. Yeah.
Great. Thanks, everyone. I will end the meeting now for all. Thank you, Mite, again for joining.
Thanks a lot. See you. Bye.
Thank you. Bye, all.